Okay, so today we are going to start talking about what constitutes a muscle cell and what are these cells doing and the parts of the cells. So movement is a fundamental characteristic of all living things. We need to move, and we do that with the capability of muscles. Muscle cells are capable of converting chemical energy of ATP into mechanical energy. And there are three types of muscle: skeletal, cardiac, and smooth. And then you have physiology of skeletal muscle, meaning we need to warm them up so that they can be quick and strong, and they have endurance, and they also have the ability to fatigue or get tired. So there are several characteristics of muscle. First, responsiveness or excitability. Muscles respond to chemical signals, and they can stretch, and electrical changes happen across a plasma membrane that causes them to react and to become excited. They also can conduct. There are local electrical changes that trigger a wave of excitation that travels along this muscle fiber that allows them to then contract or to be stimulated. Which brings us to contraction. They are able to shorten when they are stimulated. They are also capable of being extended or being stretched when they are not contracting. And they have elasticity. They return to their original resting length after being stretched. So they are able to contract and relax and stretch and, do all these different movements because of the properties they contain. So here is a picture of a skeletal muscle. Remember, skeletal muscles are voluntary. They are striated muscles that are attached to one or more bones. Voluntary meaning you're moving them. Remember, cardiac and smooth muscle are involuntary. So skeletal muscle, you make move. And they contain striations, which are alternating light and dark transverse bands. And you can see them here in this picture. And that is a result from an overlapping of these internal contractile proteins that we will talk about later when we talk about how a muscle actually contracts. Again, they are under conscious or voluntary control. And then we have a muscle cell, a muscle fiber, a myofiber, and they are as long as 30 centimeters. So we have tendons that act as attachments between muscle and the bone matrix. We have the endomyosum, which is the connective tissue around a muscle cell. We have a perimyosum, which is the connective tissue around muscle fascicles. We have an epimyosum, which is the connective tissue surrounding the entire muscle. And then we have, and that is continuous, these layers can be continuous with collagen fibers of tendons, and in turn with the connective tissue of bone matrix. Collagen is what gives our muscles the ability to be extended and to have that elasticity. It is able to stretch slightly under tension and recoil when released. It resists excessive stretching and protects muscle from injury. And then it returns the muscle to its resting length when there is no longer that stimulus to contract. It contributes to the power output, meaning how strong your muscles is, and to our muscle efficiency. So Within a muscle fiber, we have several things, and we need to know the definitions of these things. So we have what's called a sarcolemma. This is the plasma membrane of a muscle fiber. We have sarcoplasm. This is the cytoplasm of a muscle fiber. We have myofibrils. These are long protein bundles that occupy the main portion of the sarcoplasm. We have glycogen, which is stored in abundance to provide energy when you have an excessive amount of exercise. And we have myoglobin, which is the red pigment that stores oxygen needed for muscle activity. We have multiple nuclei, which are flattened nuclei pressed against the inside of the sarcolemma, and myoblasts, which are stem cells that fuse to form each muscle fiber, along with satellite cells, which are unspecialized myoblasts remaining between the muscle fiber and endomyosin. And these may multiply and produce new muscle fibers at some point. We repair by fibrosis rather than regeneration of functional muscle. And we have many mitochondria that are packed into spaces between the myofibrils. You also have a sarcoplasmic reticulum, the SR, which is smooth ER, that forms a network around each myofibril. This is a calcium reservoir, because we're going to see later that calcium activates the muscle contraction process. You also have what's called a terminal cisternae, which is a dilated end sac of the SR, which cross muscle fibers from one side to the other. We have something called a T-tubule, which is basically an infolding of the sarcolemma, which then will help penetrate through the cell and emerge on the other side, which is where we're going to see these voltages passing through, these stimuli when we go to contract. And there's something called a triad, which is a T-tubule along with two terminal cisterns. So these are all just parts of the muscle fiber, and we need to be aware of their definitions and what they do and where they are within our muscles. So we have thick filaments. These are made of several hundred myosin molecules. Myosin is the protein within a thick filament. Okay, so thick myosin. They are shaped like golf clubs. They are two chains intertwined to form a shaft-like tail. And they have a double globular head. The heads are directed outward in a helical array around the bundle. Heads on half of the thick filament angle to the left, while the other heads angle to the right. And then there is a bare zone with no heads in the middle. We also have thin filaments, which are fibrous actin. So when you hear actin, think thin. These are two intertwined strands. There is a string of globular G-actin subunits, each with an active site that can bind to a head of the myosin molecule. So the actin in this picture are the red balls. You also have tropomyosin, and these each are blocking 6 to 7 active sites on the G-actin subunits. Those are like the white bands that you're seeing here. And then we have troponin. They are small calcium binding proteins on each tropomyosin molecule, and those are the little yellow cones that you're seeing. So right now we're just going through some basic definitions because we need to understand the difference between a thick and a thin filament and what myosin and actin are and what's all involved in these structures, so that when we look at muscle contraction, we're clear on exactly what's going on. Okay, you also have some elastic myofilaments, one of them being titin, which is a huge springy protein, and they flank each thick filament and anchor it to its Z disc. They help stabilize the thick filament, and they help center it between the thin filaments, and therefore they prevent overstretching. You also have regulatory and contractile proteins. Your contractile proteins are your myosin and your actin, and they do the work. And then you have these regulators, which are the tropomyosin and the troponin. And like a switch that determines when the fiber can contract and when it cannot. The contraction is activated by a release of calcium into the sarcoplasm, and it's binding to troponin. Troponin then changes shape and pulls or moves that tropomyosin off the active sites of the actin. We're going to see then that once those sites are open, the myosin head can click in and a contraction can be performed. So here is a picture of an overlapping thick and thin filament. So you can see here they are not touching at the moment, but at some point they will. There will be a movement between the two. We also have accessory proteins. There are at least 7 other accessory proteins in or associated with thick and thin filaments. These anchor the myofilaments and regulate the length of the myofilaments. They deal in the alignment of myofilaments for maximum effectiveness. Ditrophin is one of the most clinically important. It links actin in outermost myofilaments to transmembrane proteins and eventually to the fibrous endomyosin surrounding the entire muscle cell. It transfers forces of muscle contraction to connective tissue around the muscle cell. And if you have a defect here, it can produce muscular dystrophy. It is shown as a red structure in the middle of this picture. Skeletal muscles have striations, and that is because the myosin and the actin are proteins that occur in all the cells. They function in cellular motility, mitosis, and transport of intercellular material. They are organized in a precise way in skeletal and cardiac muscle. So remember, smooth muscle does not have striations, but skeletal and cardiac do. And this is due to an A band, or which is sometimes referred to as the dark band, and the I band, which is a lighter band. So the A band deals with, it is the part of the A band where thick and thin filaments overlap, and that causes it to be especially dark. The H band in the middle of the A band is just thick filaments, and the M line is in the middle of the H band. So on this picture below, you can see all these different bands and their overlappings. And when the myosin and the actin and all these things come together, they basically start banding, or some of them are darker, some areas are darker and lighter, which is why we get these striations, why it looks to us like it's striped. So the I band is the alternating light band. And this has to do with that the bands are reflecting polarized light. You also have a Z disc, which provides anchorage for the thin filaments and the elastic filaments, and it bisects the I band. And then you have a sarcomere, and this is the segment of the myofibril from one Z disc to the next. So basically, the center of this picture is one sarcomere. And you can see that if you look at the writing here. Between the two Z discs, you have everything going on. And again, it's because of the alignment of the thick and the thin filaments and what's where that gives it this different coloration or this striping. Okay, here's another picture, kind of showing you again how you actually see the dark and the light bands here. So muscles actually shorten because individual sarcomeres are actually shortening, and they are pulling the Z discs closer to each other. So a sarcomere is the functional contractile unit of a muscle fiber. A sarcomere is the segment from Z disc to Z disc. It is the functional contractile unit of muscle fiber. Muscle cells shorten because their individual sarcomeres shorten. And the Z disc or the Z lines are pulled closer together as thick and thin filaments slide past one another. Neither thick nor thin filaments actually change their length during shortening. It's just that they are overlapping, and this causes the changes. They are sliding by one another. They're not physically getting shorter. They are just passing the other one. And then during shortening, we have these accessory proteins that also help tighten everything up and pull these proteins together. And that brings us to the end of the lecture. I know there were a lot of new terms here. Do take some time to go through them and understand the function of each of these items and where they appear within the muscle cells and how they're working together. Because all this information is going to be relevant when we talk about actual muscle contraction.